Today we will discuss two experiments that were crucial in determining the genetic code of amino acids. Amino acids are the basic building blocks for proteins. Codons, or three-letter codes, help determine which amino acids will be used during protein synthesis. The letters in the code consist of a combination of adenine, guanine, cytosine, and uracil. The first experiment that helped break the genetic code was conducted by Nirenberg and Mattaei in 1961. In this experiment, the two scientists took mRNA strands of only one of the following bases, A, G, C, or U, and added it to a mixture of components that help synthesize proteins. For example, in the initial experiment, they took polyU-tails and added each into 20 separate tubes. Each tube contained one of the 20 amino acids labeled with radioactive material. From there, they incubated the tubes and allowed them to proceed with protein synthesis. If a tube produced a radioactive protein, then the protein utilized the radioactive amino acid to complete its structure. From this initial experiment, Nirenberg and Mattaei concluded that UUU resulted in the amino acid phenylalanine. Likewise, CCC produced proline, AAA lysine, while GGG did not produce any. This meant GGG did not code for any amino acids. Nirenberg and Mattei's experiment proved crucial in demonstrating that RNA is responsible for protein synthesis. It also helped determine the amino acid sequence for 3 of the amino acids, but what about the other 17? This is where Nirenberg and Later's approach came into play in 1964. Using short mRNA strands of various combinations of nucleic acid bases, they mixed together short mRNA strands, ribosomes, and tRNAs with attached amino acids into a solution. This time, one specific amino acid in each tube was radioactively labeled. During incubation, the ribosome would bind to the mRNA strand along with its complementary tRNA strand. The solution was then passed through a filter. Unattached tRNAs filtered out. Ribosomes that binded to the mRNA strand and selective radioactive tRNA did not pass through the filter, making the filter radioactive. Through this process of elimination, Nirenberg and Later were able to identify 64 codons, meaning some amino acids have several. Tryptophan is the only one with a single codon, UGG. AUG is the start codon, but also codes for methionine. 3 of the codons, UAA, UAG, and UGA, do not represent any amino acids but are considered stop codons. These two experiments were vital in understanding the process of how proteins form by breaking their code. This code makes up every living organism, including you and me.